Welcome to this episode on the biology of appetite regulation, a subject that fundamentally challenges our traditional understanding of why we eat and why we gain weight. My name is Milan Toma. I'm an associate professor at New York Institute of Technology, College of Osteopathic Medicine. For decades, the dominant view of weight management was based on a simple, mechanical model. The concept that hunger drove eating, eating provided energy, and any excess energy was simply stored as fat until it was needed. This highly simplified view suggested that weight gain was merely the result of a straightforward imbalance between calories in and calories out. Consequently, the solutions offered often focused primarily on conscious control of food intake and increasing physical activity. However, the scientific landscape has dramatically shifted. Advances in molecular biology and endocrinology have revealed that the regulation of appetite and overall energy balance is governed by an extraordinarily complex network. This network involves intricate hormonal signals, specific neural pathways, and sophisticated metabolic feedback loops. Crucially, most of this regulatory activity operates largely below the level of conscious awareness. Before we go into these specific mechanisms, it is absolutely vital to emphasize a foundational point. Understanding why we gain weight requires a careful interpretation of scientific evidence. This evidence is derived from extensive epidemiological research and large-scale cohort analyses. We cannot draw definitive conclusions about population trends solely from any one individual's experience. The findings we discuss today represent general trends and statistical relationships observed across extensive data sets and diverse cohorts. These findings highlight patterns and associations, but they do not, and they cannot, explain or predict the experiences of every single person. Nutrition and dieting are extraordinarily complex sciences, and individual variability is the norm, not the exception. Therefore, the analyses and conclusions presented here should be viewed as informative for populations, rather than as one-size-fits-all explanations or prescriptions. To truly understand how the system works, we must first change our perception of fat tissue. Far from being passive repositories for excess energy, fat cells actively communicate with the brain about the body's energy status through sophisticated chemical messengers. The stomach, the intestines, the liver, and various other organs contribute their own vital regulatory signals to this system. All of these inputs create a biological control system of remarkable complexity. Understanding these intricate mechanisms is vital for explaining why appetite regulation can fail in certain circumstances, why some individuals are biologically more susceptible to weight gain than others, and why simple, conscious approaches to weight management often prove inadequate in the long term. We will begin by examining the surprising discovery that fat tissue itself serves as a critical endocrine organ in the regulation of appetite and metabolism. For a long time, fat tissue was viewed almost solely as a passive reservoir for excess calories, merely holding them until they were needed. Yet, contemporary research has proven that fat tissue is, in fact, a dynamic endocrine organ. It produces hormones that play critical roles in regulating appetite, metabolism, and overall energy balance. This realization that fat tissue is an active endocrine organ fundamentally changes our understanding of the system. The absence of fat tissue leads to severe metabolic and behavioral disturbances, demonstrating that its function is far more than just storage. One striking way scientists prove that fat is an active, necessary organ is through the study of rare genetic conditions characterized by a near total absence of fat tissue. For instance, in conditions such as generalized lipodystrophy, individuals may be born with a normal amount of fat, but rapidly lose it in early childhood. Despite appearing extremely lean, these individuals experience profound metabolic disturbances. They lack the normal padding provided by fat, which can result in physical discomfort and sometimes a striking appearance of sunken features. More importantly, the absence of fat tissue deprives the body of essential hormonal signals, leading to severe metabolic consequences. In generalized lipodystrophy, the relentless hunger experienced by these individuals is often described as insatiable. Because the lack of fat tissue means that excess calories have nowhere to be safely stored, these calories accumulate in other vital organs, such as the liver. This accumulation leads to liver enlargement, inflammation, and blood profiles that paradoxically resemble those seen in obesity, even though the individual is extremely thin. For example, they may exhibit high levels of circulating fat and cholesterol. The absence of fat tissue in lipodystrophy leads to severe metabolic disease, including fatty liver and abnormal blood lipids, despite extreme thinness and relentless hunger. The drive to eat in these individuals is overwhelming, reflecting a state of perceived starvation. Attempts to restrict food intake can actually provoke extreme distress and noticeable behavioral changes. The study of lipodystrophy demonstrates clearly that metabolic disease can occur in the absence of obesity. Fat distribution and, critically, hormonal signaling are just as important as the total fat mass in determining health outcomes. We must recognize that fat tissue is not merely a storage depot for energy. It is an active endocrine organ whose absence leads to profound metabolic and physical consequences. The biological basis for this complex appetite regulation system was illuminated by the groundbreaking studies involving a specific genetic mutant mouse, which scientists designated the OB mouse for obese. This mouse carried a defect in a single gene. This specific defect resulted in excessive eating and led the mouse to accumulate 3 times the normal body weight and 5 times as much fat as typical mice. The study provided concrete evidence that a single gene could profoundly influence both appetite and overall body weight. Through extensive genetic research, scientists were able to identify that the affected gene was expressed solely in fat cells. This gene was responsible for producing a hormone. This hormone was later named leptin, derived from the Greek word for thin. Leptin was found to play a vital role in regulating appetite, metabolism, and other critical biological systems. The importance of this discovery became undeniable when leptin was administered to the OB mice. Upon receiving this hormone replacement, the mice's appetite normalized and their body weight returned to typical levels. This demonstrated conclusively that leptin deficiency was the underlying cause of their severe obesity. Leptin, therefore, is a hormone produced by fat tissue that acts as a signal to the brain about the status of the body's energy stores, regulating both appetite and metabolism. It acts as a messenger between the fat tissue and the brain, primarily targeting the hypothalamus, which is the area that governs hunger and satiety. Under normal circumstances, when fat stores are sufficient, leptin levels are high. This high level signals to the brain that energy reserves are entirely adequate, and consequently, appetite is suppressed. Conversely, when fat stores become depleted, leptin levels fall dramatically. This drop in signaling triggers powerful, inherent biological drives to eat. This is precisely what occurs in individuals who suffer from a true lack of fat tissue, such as those with generalized lipodystrophy. In these cases, leptin is virtually absent. The brain interprets this lack of signal as a severe state of starvation, irrespective of the individual's actual food intake or overall body composition. This profound miscommunication leads to relentless hunger and significant metabolic dysfunction, including the fatty liver and abnormal cholesterol levels previously discussed. The state of leptin deficiency causes the brain to perceive starvation, driving excessive hunger and resulting in metabolic disease. Fortunately, for individuals with this true leptin deficiency, leptin replacement therapy offers a remarkable intervention. This therapy can rapidly reduce hunger and normalize eating behavior, often within a matter of days. While the replacement therapy does not restore the lost fat tissue, it effectively alleviates the debilitating sense of starvation. Furthermore, it protects organs such as the liver from further damage caused by ectopic fat deposition. The therapeutic potential of hormone replacement in specific disorders is clear. Leptin therapy can normalize appetite and improve metabolic health in cases of leptin deficiency, even without restoring the missing fat tissue. However, the biological picture becomes far more complicated when we consider the vast majority of individuals who experience obesity. In most people with obesity, leptin levels are not low at all. Instead, they are markedly elevated. This high concentration reflects the increased mass of their fat tissue. Following the simple model of leptin action, these high concentrations should signal to the brain, specifically the hypothalamus, that energy stores are abundant. This signal should, in turn, effectively suppress appetite and promote increased energy expenditure. Yet, in obesity, this vital feedback loop breaks down. Despite the abundance of leptin, the brain becomes significantly less responsive to its signal. This phenomenon is known as leptin resistance. Leptin resistance presents a fundamental paradox. The body possesses ample, even excessive energy reserves, given the high fat mass and high leptin levels. However, the brain behaves precisely as if the body were in a state of severe deprivation. The hypothalamus, unable to properly hear the strong leptin signal, fails to adequately suppress hunger. This failure actively continues to drive food-seeking behavior. Simultaneously, the body's energy expenditure may be reduced. The system attempts to conserve what it mistakenly perceives as limited resources, further cementing the weight gain process. This profound mismatch between the body's actual energy stores and the brain's mistaken perception of starvation creates a powerful, relentless biological drive to eat, even in the physical presence of significant obesity. The origins of leptin resistance are highly complex and multifactorial. It is not a single, simple mechanism. Chronic overnutrition plays a role. Inflammation within the hypothalamus itself is a contributor. And disruptions in intricate cellular signaling pathways all contribute to the brain's diminished sensitivity to the leptin hormone. As a direct result of this resistance, simply increasing leptin levels, whether this occurs naturally through further weight gain or by medically administering the hormone, does not effectively restore normal appetite regulation in most people who live with obesity. This is a key distinction. Unlike the individuals with true leptin deficiency who respond dramatically and rapidly to leptin replacement, those with leptin resistance experience very little or often no reduction in hunger or weight when they are given additional leptin. This state of leptin resistance is central to explaining why achieving weight loss is so notoriously difficult, and why maintaining that loss is an even greater challenge. When an individual successfully loses fat, their circulating leptin levels naturally fall. Importantly, the brain's underlying sensitivity to leptin does not automatically improve. Instead, the drop in leptin, even if still higher than a lean person's baseline, is interpreted by the resistant brain as a serious threat to energy balance. This perception triggers powerful physiological adaptations. The brain initiates increased hunger signals and simultaneously decreases the body's energy expenditure. These powerful biological adaptations persist long after the initial weight loss has been achieved, serving as a powerful mechanism that promotes weight regain. Sustained weight reduction thus becomes a formidable challenge, fighting against a biological system that is mistakenly convinced the body is starving. Thus, the key takeaway is this. In obesity, high leptin levels fail to suppress hunger because the brain has become resistant to leptin's signal. This leptin resistance causes the brain to act as if the body is starving, driving persistent hunger and reduced energy expenditure despite the actual presence of abundant fat stores. The discovery of leptin fundamentally transformed the entire scientific understanding of both obesity and appetite. It demonstrated conclusively that body fat is not merely a passive consequence of overeating. Instead, fat tissue is an active participant in a sophisticated feedback system that regulates overall energy balance. The implications here are profound. Obesity and leanness are not determined solely by willpower or simple lifestyle choices. They are influenced by complex hormonal signals that possess the power to override conscious control and choices. We must appreciate the deep biological basis of appetite and weight regulation. Hormonal feedback systems originating from organs like fat tissue are powerful drivers of eating behavior and overall energy balance, not just conscious choice. The regulation of appetite and body weight is intrinsically governed by hormonal signals emanating from fat tissue, with leptin playing a central, critical role. Disruptions in this delicate system, whether those disruptions are caused by a specific genetic mutation or by the catastrophic loss of fat tissue, can lead to profound metabolic and severe behavioral consequences. In rare but illuminating cases of true leptin deficiency, restoring the missing hormone can correct the underlying drive to eat, strongly highlighting the biological basis of appetite and the potential for targeted therapies in specific forms of metabolic disease. We must recognize that fat tissue is an active endocrine organ. Its absence leads to severe metabolic and behavioral disturbances, not simply thinness. we must understand the central role of leptin in appetite regulation. Leptin signals the brain about energy stores, suppressing hunger when fat is sufficient and triggering hunger when it is low. We must appreciate that metabolic disease can occur entirely in the absence of obesity. Conditions like lipodystrophy clearly demonstrate that fat distribution and the state of hormonal signaling are just as important as the total fat mass itself. Finally, we must consider the biological basis of appetite and weight regulation. Hormonal feedback systems, and not just conscious choice, are the ultimate drivers of eating behavior and energy balance. The bottom line is that fat tissue and its hormone leptin are central to the regulation of appetite and metabolism. Disruption of this system, whether by a genetic mutation causing deficiency or by physiological changes causing resistance, leads to severe metabolic disease and relentless hunger, fundamentally underscoring the deep biological complexity of weight regulation. This is why the journey to understanding and managing weight is a biological one, far beyond the simplified calculations of calories. I hope our discussion has illuminated the critical final point, that the regulation of appetite and body weight is intrinsically governed by complex hormonal feedback systems, with leptin playing a central role, and not solely by conscious choice or willpower. The science demonstrates that fat tissue is an active endocrine organ, and understanding the biological complexity, including phenomena like leptin resistance, is vital for appreciating why weight management is such a profound challenge. Thank you for joining me today on this exploration of the biology of appetite regulation. If this episode on the biological mechanisms that drive hunger and satiety has sparked your interest, you may wish to explore more in one of my books.